Welcome back, everyone. We were looking at enzymes. Enzymes were a specific, special case of globular proteins that we learned about in the last unit. Enzymes, we found, were biological catalysts. They take care of catalyzing any and all biochemical reactions that are required within biological systems. And what we had left with were the 6 classes of enzymes, 6 classes, not names, but classes of enzymes that typically carry out these types or catalyze these types of reactions. They can be put into 6 categories. We had gone over in detail the names of them and examples of these 6 classes of enzymes, and we left off there. And what I'd like to do now is take a sort of real-life look, a specific look at a certain class of enzymes, that of the hydrolase class, one type of enzyme that we're going to be looking at, just to show that we can show classes and we can talk about theory and everything, but these actual enzymes in real life belong to these classes. So let's take a look at that. The type of enzymes in this hydrolase class we're going to be looking at are called proteolytic enzymes. Proteolytic enzymes, we'll be looking at 3 in particular, 3 representative proteolytic enzymes. There are plenty more of them, but ultimately proteolytic enzymes are digestive enzymes. They're within our gut as we take in foods and proteins and starches and that sort of thing. Specifically, proteolytic enzymes are tasked with taking our proteins, which come in in a dietary manner, and breaking them down into their individual amino acids. Remember, proteins are a polymer, polymers of amino acids, and we utilize those amino acids to make our own proteins. But however, all of the proteins and the polymers that we take in a dietary sense need to be the peptide bonds which connect those amino acids need to be hydrolyzed, and thus they are a hydrolase class of enzymes which cleave amide bonds, the bonds between these amino acids, at specific amino acids. Remember, there are 20 different amino acids, and different proteolytic enzymes are specific for cleaving, breaking that peptide bond at a particular amino acid. So if we look down here on the bottom, we have a tripeptide. Remember, a peptide is like a small protein of 10 amino acids or less. We have alanine, phenylalanine, glycine. Down here on the bottom is written out in their 3-letter abbreviations with these dashes between them. That indicates the amide bonds that ultimately connect them. And up above it is the actual structure of these amino acids within the peptide. Now notice, what if we had a proteolytic enzyme which specifically cleaved at phenylalanine on the bottom? So if you'll notice, phenylalanine now has, if you look to the right and to the left of that phenylalanine side chain, it actually has two amide bonds associated with it. So if you look to the left of this, here's the phenylalanine side chain right here. If you look to the left, there's an amide bond, and if you look to the right, there's an amide bond as well. So what proteolytic enzymes do is they hydrolyze let's see here, the peptide bond on the right-hand side of that side chain. It's considered the carbonyl side, if you will, but it's always on the left-hand side. So if we say, all right, here is our amino acid that is going to be hydrolyzed by a particular proteolytic enzyme, we always look to the peptide bond at the right of it. That's the one that will get cleaved. The one on the left will not be cleaved by the proteolytic enzyme. So always look to the right when we do this. So there is a particular name for one of our three proteolytic enzymes that cleaves at phenylalanine and a number of other ones. So let's take a look at that. Well, the first class of our three proteolytic enzymes. So the first one is chymotrypsin. Chymotrypsin is specifically cleaves at aromatic side chains. Side chains that happen to have an aromatic ring on them. And there are three amino acids which do that: phenylalanine, tryptophan, and tyrosine. Those three. If you look just below here, we have pictures of those. There they are. You can see that the phenylalanine has the aromatic side chain or the aromatic ring on all of them, so that we see that these would be cleaves. So we keep those in mind. Chymotrypsin cleaves at phenylalanine, tryptophan, and tyrosine. Let's move on to the second representative class of proteolytic enzymes. The next one would be known as trypsin. Now, trypsin is specifically cleaves at basic side chains. Basic. Now, basic means, now remember, amines are basic, are natural bases. So some of our side chains have amines or amine type structure on them, notably lysine and arginine. Down below, we have the structures of our lysine and arginine showing their specific side chains. So if we had the proteolytic enzyme of trypsin, that would cleave at those two amino acids. Let's look next at our third representative class of proteolytic enzymes. That's known as elastase. Elastase is specific for very small side chains, notably only glycine and alanine. Remember, they're the smallest amino acids as far as the side chain is concerned. Glycine has just a lowly hydrogen for a side chain, and alanine has just a methyl group. Everything else is bigger than that. So thus, elastase, if a peptide chain is treated with elastase, it will cleave only at alanines and glycines, and specifically at the amide bond to the right of that side chain. So let's look at an example of this. Rather than showing you a complete structure of, say, an 8 or a 10 amino acid peptide sequence, I'm going to abbreviate that to what we see here. And what we see here is 1, 2, 3, 4, 5, 6, 7, 8. An 8 amino acid peptide, and we have our three-letter abbreviations here for all of these amino acids, glycine, phenylalanine, arginine, alanine, and so on. So, and notice that the amide bonds that connect these in abbreviated fashion are given as these dashes. So it's easier to write on a drawing program or something like that. What these dashes are the amide bonds that connect these amino acids into our polymer, into our peptide. So the question here now is, what if we treated this particular peptide with chymotrypsin? Chymotrypsin, if you remember, specifically cleaved at aromatic amino acids. And here we have one. There's tryptophan is right here, so it's going to cleave at this amide bond right here to the left. It's the only one on tryptophan, so it's to the right, not left. Also, phenylalanine here. Now notice phenylalanine has a peptide bond on the left and on the right. It always goes for the right, so we cleave there. Lastly, tyrosine is also under the auspices of the amino acids in a peptide which are cleaved by chymotrypsin. Again, it has or at least a representation of a peptide bond on the left and the right, but we always go to the right, and that gets cleaved here. So let's see what we would get out of this if we treated that 8 amino acid peptide with chymotrypsin. Well, we'd get 4 fragments here based upon the peptide bonds that we cleaved. Now, interestingly, there's a couple ways we could ask a question here. We could ask the question, all right, here if we look at the 8 amino acid peptide up top, we could say which bonds will get hydrolyzed by chymotrypsin, and we already saw those. Or we could ask how many fragments would form as a result of treating this 8 amino acid peptide with chymotrypsin, and that would be a different answer. And if we look, here we go. We have, well, let's see. We have 1 free amino acid tryptophan, 2, 3, and 4. So it would be 4 fragments, 2 of which are just free amino acids. But there's a number of ways a question could be asked if this were a test or a quiz or an exam on that. So let's move on here. Let me actually clean this up a little bit, we'll move on, and we'll take that fragment profile that we see that's visible right now, and we're going to treat that with trypsin. You can probably see where this is going. Trypsin, okay, trypsin cleaves at basic side chains, notably lysine and arginine. So if we had to, so there's actually only one peptide bond, and it happens to be the one on the right. So this is going to get cleaved here, and we're going to get cleaved here. And let's look at the product profile we would get from that. And there it is. So we would have this product profile based upon the cleaving of those 2 amino acids. So here now we have it's got a bunch of free amino acids, and we have a couple of dipeptides left over. And you can probably guess where we're going to go from here. We're going to take this series and treat what's left here with elastase. Shockingly, there's only a glycine and an alanine left to cleave on here, and if we look very, very closely, we're going to cleave at glycine right here, alanine right here, to give our final grouping, which are just all free amino acids. This is very fanciful, and it's just written to show that, how this series would take place. This wouldn't really happen in real life, but it gives an idea of how we can, what sort of questions could be asked on this topic, and so on and so forth. Now, I've blabbered on with a whole lot of who shot John about how enzymes catalyze these reactions, but we've never really talked about the practical way this is carried out. What physical process is carried out when an enzyme catalyzes a reaction? What's exactly going on? Well, the enzyme itself is, so we have an enzyme, and then we have a substrate. The substrate is the molecule which an enzyme is catalyzing, or catalyzing a reaction of. Now, so the first thing we have to realize that the enzyme and substrate must be in physical contact. Physical contact, that is important. And also, if you look at the diagram here, typically, and this is just, again, a very representative, fanciful drawing, typically an enzyme is an orders of magnitude much larger than its preferred substrate, as given in this representation right here. It's much bigger. But where the action happens on the enzyme is in what is called the active site. The active site is a sort of an indentation on a particular part of the enzyme in which the substrate fits very nicely. So we have that. So the enzyme is very large, but it's only one very small part of it which actually comes in contact with the substrate. The rest of that enzyme is sort of supporting that active site where the actual chemistry takes place. Now, the substrate must bind specifically at the enzyme's active site, and the chemical reaction moves forward. Let's harken back to our proteolytic enzymes example, specifically with chymotrypsin, if you will. And chymotrypsin was specific for amino acids who had that had aromatic rings on their side chains. Aromatic rings have a specific shape to them, and ultimately what happens is the active site of chymotrypsin has a topography which can accept very readily an aromatic ring, or an aromatic ring built into the active site. Specifically, in this diagram here, we have a representation now of so this what we see right here on the diagram, this could be the binding pocket or the active site, if you will, of chymotrypsin. And notice that it fits like a glove with that aromatic ring in there. It's specific. We have a specific fit, like a hand in a glove, for that aromatic ring. If other amino acids sort of came in there, like an alanine or any other one that didn't have that type of side chain, it would try to fit into that binding site, and it would just get kicked right back out again because it doesn't have the exact fit. Here now, with this particular side chain, we have a very good fit and thus a very tight fit. So ultimately, so we have that phenylalanine fitting in there, and then we get a very secure binding of that phenylalanine into there. Once we get that secure binding, so this phenylalanine is caught in there and not moving, then we have something called the catalytic domain. And this protuberance right here on this representation, that's where the actual chemistry happens. The catalytic domain are almost always amino acid side chains which actually have this chemistry being carried out. And in this case, that chemistry would be breaking this amide bond right here on this particular amino acid. Again, all other amino acids just don't get held into that site, so it is only amino acids whose side chains have an aromatic ring on them that we get this enzyme-substrate complex to form. And the enzyme-substrate complex is a very strong binding, physical contact between a particular, in this case, amino acid side chain and the active site of the enzyme. Here's just an example of an X-ray crystal structure of a representative globular enzyme, globular protein enzyme, with its substrate bound in the active site. Now, we don't really need to talk about what enzyme this is. They have a name down on the bottom there, what exactly it does, but this is just to show a number of general things. The first thing over here on the left that I'd notice is, notice the size difference between the actual enzyme itself and the substrate which is bound within it. There's a huge difference in size there. If you look at this enzyme, it's put into ribbon structure that we got sort of used to in the last unit. We can see in here we have units of secondary structure and ribbon structure. There's an alpha helix, beta sheet right there. We have some random loops up here. So it's your standard structure of a globular protein, but within it, it does have that very important active site right there, in which this particular substrate fits very, very readily, and it fits tightly and readily, and thus we once it's fit very tightly like that, the actual chemistry, whatever that might be on this particular example, can indeed take place. So we're going to look now at specifically at the way in which, or models in which the substrate binds in the active site of the enzyme, because we have to have a scientific model behind it. Now, up to, for probably around a century, there was one particular binding model, if you will, of the enzyme-substrate complex, and we'll take a look at that next. This particular model was known as the lock and key model, and that had some currency. If we think about a lock and a key, a lock and a key are made to fit exactly. There's one lock and there's one key, and that only one key can go into that lock to unlock the door. All other keys do not fit in the lock, and that's analogous for a particular substrate fitting very snugly, well and tightly into an active site, and all other substrates will not do that. So that lock and key method, and if we look down here on the bottom, we see a representation of an enzyme and a substrate, and here our substrate has a particular topography associated with it, and the enzyme, if you'll notice, its active site has the exact reverse topography, again, sort of like a key in a lock, and they're made to fit exactly together. And if we look over here on the right now, we have that very tightly bound enzyme-substrate complex. Now, this model worked very well for many years. It worked working with enzyme kinetics, for working with explaining how well a particular substrate bound to an enzyme. But with the advent of technology, the advance of technology, different instrumentation, we were actually able to gain through typically through X-ray crystallography a comparison between the topography of the active site in the unbound enzyme and the topography of the active site in the bound enzyme, and those were found to be slightly different. So we had to update our model. So the lock and key model fails to take into account protein in the active site, conformational changes to accommodate the substrate molecule. They're not the same. So we had to update this model somewhat. And what we fell into was the currently accepted model, which is known as the induced fit model. So we don't think of the lock and key model anymore as the up-to-date version of the type of a model of enzyme substrate binding we want to look at. It is now the currently accepted model is known as the induced fit model. Now notice here, what we have going on is our enzyme and substrate over here on the left are the topography of our substrate now, and this fanciful representation is similar but slightly different than the reverse topography of the enzyme's active site. Notice on the substrate there's sort of jaggy lines there, they're corners and that sort of thing, but not so on the actual enzyme. And what ultimately happens is that the enzyme active site has enough of a contour to it to initially accept the substrate, but once the substrate is bound to it, it alters that topography to better accommodate that enzyme. Notice this active site now has jaggy corners on it. It's not very loose topography as we saw over here. So we get an initial binding of that substrate, but then that induces the active site topography of the enzyme to change. So that's why we call this the induced fit model. So we are progressing our discussion here. First, we talked very, very blandly and generally about an enzyme catalyzes a chemical reaction of it going from a substrate to a product. But then we asked the question, well, how exactly does this happen? And we said that, well, there has to be physical contact between the enzyme and the substrate, and thus getting very strongly bound enzyme-substrate complex, which we see here. But notice, no chemistry has happened yet. All we've managed to do is to get a very tightly bound enzyme-substrate complex. But once we had alluded to the fact that once this substrate is strongly bound to the enzyme, the chemistry happens. And what does that ultimately mean? Well, in this example, let's look at this particular substrate right here. This could be, it looks like it's sort of a dumbbell you see at the gym, but what you might consider it being is like one part of a molecule here, another part of a molecule there, and then maybe a bond, that skinny part on the dumbbell could be thought of as a covalent bond. So we'll look at that as a covalent bond here, just for this representative example. And over here now, we have that molecule with its attendant covalent bond bound very strongly in the enzyme's active site. So what ultimately happens now is the chemistry takes place, and that happens within the catalytic domain, within the active site of the enzyme, and we go from substrate to product in that direction. So at some point, we're not the substrate anymore, we have become the product, and we're going to look at that midpoint. that if you can imagine a midpoint between the change between a starting material and a product. This is sort of an ethereal concept and a little bit hard to think about. But this is what we would term the transition state in an enzyme-mediated reaction, a transition state, and that's given by, so notice right here on this fanciful design that that bond here that we saw, the representation of a covalent bond is bending. There's no such thing as a bent covalent bond, but so this is again a very representative view of that. But what we see here is a transition state. And notice up here on the top, we've been going enzyme plus substrate, E plus S, into that ES, enzyme substrate complex. And we show the transition state as like an ES star, if you will, whatever that means. It means we're at the transition state. And if you look, if we had to put a definition to it, a transition state is when whatever is bound within the enzyme, within that binding pocket, has features of both the substrate and the product. It's like a halfway point in the reaction, again, shown by the bent covalent bond, which is ridiculous. There is no such thing as that. But that's the halfway point. That's how we're representing it. And at some point, we're done with that halfway point, and we have a product formation. And notice here, well, this is the enzyme product complex. Enzyme product complex. So it's not the enzyme substrate complex anymore. It's the enzyme product complex because we have in here the product of the reaction. Now notice in this product shows that covalent bond is broken now. If that happened to be the particular role of that enzyme, this is only one particular example, then we would have an active site stuffed full just of product. Importantly, what ultimately happens is, whereas the enzyme substrate complex needed to be strongly bound, the enzyme product complex must be weakly bound. It must be. If that product was strongly bound within that enzyme's binding site, that binding site would never get freed up to spit out the product. It would just completely stay in there. So ultimately, if we have a very weak binding on this enzyme product complex, this weakly bound sort of interaction, that product is going to diffuse out of the enzyme. So we have just the enzyme and just the product. So they're separate again. Now notice we have freed up the enzyme. This enzyme over here on the right is the same thing as the enzyme over here on the left. It has not changed. So thus, that same copy of the enzyme can cycle around again and pick up another substrate here and have the process go cyclically now, over and over and over. This is why we only need a tiny catalytic amount of our enzyme, just a few copies, to turn many substrates into products, because we have this what's called a turnover, or this cycle. The same enzyme is catalyzing substrate to product conversions over and over and over. Now, a way of representing how efficient an enzyme is through something known as the turnover number. The turnover number is defined as the number of substrate to product conversions just one copy of an enzyme can carry out before it becomes destroyed or inactivated or used up, and that sort of thing. Typically, turnover numbers on the order of 100,000 to 1 million are not unusual for just one copy of an enzyme. So it is truly catalytic in nature. So let's try to bring this together. We'll talk about the types of chemical reactions that enzymes tend to catalyze. And because these types of reactions have a bearing on what the active site, or rather what the transition state looks like, we have three common transition state features that we want to look at, which represent the most common types of enzyme-mediated reactions that we're going to be looking at. The first transition state feature, really the first type of reaction we're going to be talking about, is a reaction which breaks a covalent bond. We had just looked at that in the last slide, but we're going to take a closer look now. We're just going to define it here. There are first of three transition state features would be breaking a covalent bond, which has to orient a substrate in the active site specifically to place stress on that bond. Look over here at this structure on the left, and we have this protuberance here that is placing stress on that bond right here. And we saw in the transition state that stress becomes such that we're at a midpoint of breaking in that carbon-carbon covalent bond until finally that bond is broken. So for any chemical reactions or enzyme-mediated reactions that require the breaking of a covalent bond, how we can look at this mechanistically from the standpoint of what an enzyme ultimately does is it orients the substrate within that active site to place stress on that bond, making it weaker and disposing it towards breakage. Remember that transition state, we haven't broken the bond yet, we're disposing it towards breakage. We're using sort of flowery language there to describe the transition state because it's very difficult to describe a transition state. No such thing as a bent covalent bond, so it's being disposed towards breakage, if you will. Now let's look at our second transition state feature, and we're going to find that it is literally the reverse of the one we're looking at here, where we broke a covalent bond. Well, what about one where we're going to form a covalent bond, like a ligase class of enzymes? So the second transition state feature that we're going to be looking at now is literally a reverse of what the diagram on the other slide said. Literally a cut and paste of it reverse. So the enzyme-substrate complex puts two molecules within that active site over here on the left-hand side, and the enzyme active site works it such that we dispose both of these substrates towards each other. It literally puts them together, forces them right up into each other's grill, and it poises it in a favorable orientation towards bond formation. We take all of the guesswork out of it, all of the randomness and collision theory and everything like that that we have in determining what the rate of a chemical reaction is. We take all that out because we have an enzyme which literally forces the two covalent structures right on next to each other so that that bond can start to form here in the transition state, and over here in the enzyme product complex it has completely finished forming that covalent bond. The last one. The last most common transition state feature. Now this covers a lot of reactions, but I want to harken back to our organic chemistry and think about the many reactions we looked at which had catalysis as part of it. Catalysis specifically acid catalyzed. So we had the ester formation, ester hydrolysis, acetal formation. All these different types of reactions needed acid catalyst. So it's a catalytic acid to allow the reaction to proceed. Now, obviously, inside the cell or in the bloodstream or anything like that, we can't just acidify the entire surroundings. The cell would die, the bloodstream would become acidified, the organism would die. So what enzymes can do in this third transition state feature is it can modify the pH environment, we call it a microenvironment, just within the active site. Not all around, but just within the active site. And how might we ultimately do this? Well, if we look at an enzyme with a bound substrate, and specifically we're looking inside the active site here. So here is our active site, and so let's say we have a substrate whose conversion to product depends on having a catalytic acid environment. Well, let's think about some of our amino acid side chains. Some of them, notably aspartate and glutamate, had carboxylic acid structures in their side chains. So what if we had an active site in which we had aspartate or glutamate side chains poking into that active site? So here's a carboxylic acid. Here's another carboxylic acid side chain. They are acidic by nature. They are acidic. So thus, what happens is we get a low pH within that active site. Just for review, a low pH means acidic. High pH would mean basic. So notice that low pH, that catalytic acidic environment, is only within the active site. It is not within the surroundings. So we call that a pH microenvironment just within that active site. We create an area of low pH acidic conditions which can increase the rate of conversion of this substrate to its product. Now, the other side of the coin on that is, what if there was a reaction which required, organic reaction, biochemical reaction, which required basic conditions, not acidic conditions? Well, there are a lot of reactions which require that in the test tube, and the same types of reactions have to be carried out in biological systems. Well, let's think again about our representative amino acids, our 20 amino acids. Some of them have amines on their side chains. Amines are weak bases. Notably, lysine has a primary amine, for instance, on its side chain. What if now we had an active site which had a bunch of lysine side chains which were sort of lining that active site, and then we had a bound substrate, that substrate requiring catalytic basic conditions in order to convert to product? Well, we have that, and we have that in a microenvironment. That high pH, which means basic conditions, are only within this active site and not within the surroundings. In the surroundings, we have physiological pH. Everything is fine. It is just within the active site now that we have these, what would be considered very harsh conditions if they were exhibited within the surroundings around this enzyme and within the cell and within the bloodstream. So our last word on enzymes. Again, we keep building on this. We need a little bit more and a little bit more each time to really give a good overview of enzyme chemistry. And here's the last piece. Up to this point, we've talked about all that is needed for an enzyme-mediated reaction to occur is to have our substrate and our enzyme come into physical contact. And then we go through the transition state, go to product, the product is released, and we have our turnover again of that enzyme coming forward and picking up another substrate. And that is true with many, if not most, enzyme-mediated reactions. All that's required is the enzyme. But sometimes something else is required. It's not just the enzyme and the substrate have to bind together. Sometimes we have a third thing that needs to bind with that enzyme and substrate, and that is known as a cofactor. A cofactor. Some enzymes require other components to bind simultaneously with the substrate to form that enzyme-substrate and cofactor complex in order for a successful reaction to occur. So anything that has to have a concurrent binding with the enzyme and the substrate is known as a cofactor. Typically, they are either metal ions or small organic molecules. We're going to find that many of the metal ions that you need, that are necessary for us, for animal systems, for micronutrients, are cofactors in enzyme-mediated reactions. Like iron, zinc, magnesium, copper, typically are used as cofactors in these certain reactions which require that third piece in there. Now, the small organic molecules which we require, in addition to sometimes metal ions, are typically we're going to be seeing many of these are vitamins, vitamin A, our B complex, vitamin C, and so on and so forth. These small organic molecules are required as cofactors many times, not always, but many times in these these enzyme-mediated reactions. So let's look just at a very sort of fanciful design here, how that might be put across. So if we look at the series on the top here, we have an enzyme with its active site, and we have a substrate. And just those two together, if we put them together, will not form an enzyme-substrate complex. So thus no reaction occurs. However, if we take that enzyme and the substrate and we throw in a cofactor, in this case a copper ion, doesn't have to be that, but we have that copper co-binding with the substrate, well, then we get the formation of our enzyme-substrate complex to form a tight binding, and now the reaction will occur. That wouldn't have happened in the absence of a copper ion. So that's an example, a fanciful one, but an example of a cofactor that is required in this successful binding of an enzyme and a substrate. But we will now look at small molecules that are also sometimes required as cofactors in place of these metal ions. I'd use the example of vitamins, and we'll look specifically at the B complex of vitamins. If you look on your vitamin pill container, we have your B complex, B1, B2, B3, B6, B12. These are all cofactors which facilitate enzyme-mediated reactions within the body's energy-producing pathways, almost exclusively in energy-producing pathways. Virtually every chemical reaction that happens within animal systems is enzyme-mediated, and these reactions which produce the body's energy, which we'll be talking about in a future chapter, a future unit of study, are very important. Three of these cofactors, these small molecules I show right here, vitamin B1, vitamin B2, vitamin B3, they have their actual names to them: thiamine, riboflavin, and niacin. And importantly, now they all have a function. This function over here, it's not that important what the function is. We'll be talking about that in future units of studies. But notice that the actual vitamin is not the cofactor. And we'll see that on the next slide. The vitamin, we'll look at the structure, has to have covalent structure added to it from the biological system to actually become the cofactor. Thiamine, which is vitamin B1, has to become thiamine pyrophosphate, whatever that is. We'll take a look at that. Riboflavin must be augmented structurally to become what is called flavin mononucleotide or flavin adenine dinucleotide. We'll be talking about that, and so on and so forth. So let's look at these three vitamins right now, or three B complex vitamins, and then we'll look at the type of structure that is required for their active form, as their active cofactor. So here we are. Here's thiamine, riboflavin, and niacin. Niacin, also known as nicotine amide, because it's based on the structure of nicotine. And here they are. So this is what is vitamin B1, vitamin B2, vitamin B3 in our vitamin pill. If we take a vitamin pill in the morning or something, so we intake that into our diet, and the body utilizes these structures, but they add onto it to become the actual cofactors. And so thiamine actually becomes what, as we said, thiamine pyrophosphate. The body tacks on a couple of phosphate groups, and this is the actual cofactor that is required in that enzyme-mediated reaction, not the original vitamin. It has to be structurally augmented. Now, let's see, riboflavin. Riboflavin is this structure that we see up top here. Ribo, because it has ribose, there's a ribose sugar right there, and this fused three-ring system is known as a flavin molecule, so it's called riboflavin. And what the body has to do in red here is tack on some phosphates, tack on a sugar, and this nitrogenous base right here. And then we have the cofactor. That cofactor is what is required in the vitamin B2 is required for. It's known as flavin adenine dinucleotide, long word. So ultimately we go to the acronym FAD. FAD is going to be a very important molecule a few units of study hence when we start looking at these energy production pathways. Nicotine amide or niacin, vitamin B3. There it is. This is what we intake, and over down here the body has to tack on a whole lot of other stuff in order to bring it into its active cofactor form, which is called nicotine amide adenine dinucleotide. Again, too long. So we always go to the acronym NAD. There's a little plus right there, because ultimately if you look very closely on this structure right up on top, there is a positive charge on a nitrogen, so the whole molecule has a plus charge on it, but you don't say NAD plus, you just say NAD. We'll be looking at particularly NAD and FAD down the road in these energy production pathways.